Chapter 5 will be extensions and modifications of the very basics that we learned about Mendel. So far we have learned about very simple cases where genes have one dominant allele and one recessive allele. And the reality is that that is not always the case. In this first lecture, we will go into variations that involve one gene, and that will include this list shown in number 1, and also how sex can influence the inheritance and the expression of alleles. So this looks like a bit of a laundry list of a lot of different things, and we're just going to go through them rather rapidly, and then we'll spend some time in class trying to practice distinguishing between them. So first of all, variations involving one gene. We'll start off with incomplete dominance. So incomplete dominance is when the heterozygous phenotype is actually sort of halfway between the two homozygous phenotypes. An example is shown here with eggplant. So if you have this dark purple eggplant, it's big P, and a white eggplant is little p. So if you would cross those two together, you would get a heterozygote, and it turns out that the heterozygote is about halfway between the two. Incomplete dominance gives this characteristic ratio in the F2 generation of a 1 to 2 to 1. So this would be crossing two heterozygotes together. And here's the Punnett square shown. And so if you're homozygous, you have the one extreme phenotype or the other extreme phenotype. If you're heterozygous, you have that intermediate phenotype. Codominance is a little bit different from incomplete dominance in that the heterozygotes express both of the homozygous phenotypes. With certain things, it's really impossible to have a codominant phenotype. So for example, a pigment color is going to be really hard to show both white and purple at the same time, for example. However, some things you can simultaneously show two phenotypes. And one example is shown with the ABO blood type system, which we've previously touched on. So there are two dominant alleles. It's called big I A and big I B. Those are two dominant alleles, and they are codominant with each other. Now, the idea of this blood type is that your red blood cells have a antigen that is expressed on the surface of the cell. And so if you have the big IA, then you make this certain pink antigen on the surface of your cells. If you are B, you make a different antigen on the surface of your cells. Individuals who have type AB blood, which they have one of each of those codominant alleles, actually show both phenotypes. They show both of the antigens on the surface of their red blood cells. Incidentally, the recessive allele, the little I, which gives type O blood, actually you have no antigens on the surface of your red blood cells. Next, we'll talk about multiple alleles. So most examples we've talked about so far, we've simply had one dominant allele and one recessive allele for that gene. And it turns out that that is really an oversimplification. Most genes have many different alleles that are present in the population. Of course, one individual is only able to have two alleles at one time, but in the general population, you may find different alleles and different combinations of those alleles in different individuals. Now, one example we just spoke about actually, so the ABO blood type gene is an example of a gene with three alleles. We have the big IA, big IB, and little I, like we just talked about. I'll talk about one other example, which is a gene that influences the feather pattern on ducks. So here there are three different alleles, and those are called big MR, just plain big M, and then little md. Let's first look at the homozygous phenotypes for each of these so we have an idea of what they are encoding. So the big MR is actually called a restricted phenotype. That is shown by this brown color over here. And then if we have big M, that is called mallard, and mallard feather pattern is shown in this individual. And then we have little md codes for dusky, and dusky is this black and gray phenotype. Now, if we look at the different heterozygous combinations, what we can determine is that there is actually an order of which allele is dominant over which other allele. And so if we have the restricted allele together with the mallard allele, it will show the restricted phenotype. And so what that says is that restricted is dominant over mallard. If we look at some other combinations, so the little md, what we find is that dusky allele is recessive to the restricted allele. The dusky allele is also recessive to the mallard allele. And so we can actually make what's called an allelic series. We can determine the order of dominance between these three alleles. And so we would say that the restricted allele is dominant over mallard, mallard is dominant over dusky. And I just want to point out that this highlights a different aspect of dominance and recessiveness than we have touched on before. So specifically, the idea of dominance, it's all about which other allele you're together with in an individual. So for the mallard allele, in some cases, it will act like a recessive allele when it's together with the restricted allele. Other times, it will act like a dominant allele when it is together with the dusky allele. Next, I'd like to talk about lethal alleles. So in this case, the homozygote does not survive. And one example of that is a dominant mutation that actually causes mice to have this yellow coat color. And so we're going to call this big Y, and little y would be the normal wild type allele. In this case, the wild type is a white mouse. When we cross these two individuals, what we get, we would expect 1/4 of the individuals to be big Y, and then 1/2 of them to be heterozygous, big Y, and 1/4 of them to be little y. And so it turns out that in this particular case, the big Y never survives until adulthood. And so this is a lethal allele, and what you'd see are the other two. And so we see this characteristic ratio where you have 2/3 of the progeny are yellow and 1/3 are non-yellow or white in this case. That 2-to-1 ratio is very characteristic of lethal alleles in which the homozygote does not survive. I'd also like to mention that some alleles cause reduced viability of homozygotes. And so this is called semi-lethality. And one example of this is actually what we are doing in laboratory this semester, the cross-veinless mutant. What many people notice in their F2 generation is that they get many fewer cross-veinless mutants than they expect. And it turns out that cross-veinless over cross-veinless, the homozygotes have some reduced viability. Varying penetrance or expressivity is something else that we also have to deal with in real-life genetics. So up until now, we've assumed that every individual who has a particular genotype will show the phenotype that is expected based on that genotype. And this is not actually always true. And so there's something called incomplete penetrance, where some individuals who do have the genotype do not show the expected phenotype. There's also something called varying expressivity, where individuals with the same genotype have more or less expression of that phenotype. So an example of incomplete penetrance is polydactyly, and we know now that polydactyly is actually dominantly inherited. However, only 70% of the people who have that dominant polydactyly allele will actually show the polydactyly phenotype. And so this ends up giving these really complex looking pedigrees which don't really agree with what we would expect knowing this allele is showing dominant inheritance. Varying expressivity, an example is shown here. So if a beagle has this dominant piebald allele, it will have a spot on its back. But it varies tremendously how big that spot actually is. It can encompass almost the whole body, or it can have just a couple of very small spots. So this is varying expression of the phenotype. If you compare and contrast penetrance versus expressivity, Penetrance is looking at whether you have that phenotype or not. And so here, maybe all these individuals have the allele where you would expect them to show a particular phenotype, but only a fraction of them actually show the expected phenotype. Varying expressivity is how strong or how weak that expression of the trait actually is. And of course, you can have both of those occurring at once. Some alleles are also sensitive to temperature, and so I'll just show you two examples here. One example is found in fruit flies. There is a mutation called vestigial, which causes these little stubby wings, and it turns out that the wings are stubby if you grow them at about room temperature. This is Celsius. However, if you grow them at a higher temperature, they show normal wings. So that is a temperature-sensitive allele. Also, in rabbits, the Himalayan phenotype, if you grow these rabbits at lower temperatures, they have darker points, darker ears, nose, and feet, whereas if you grow them at a higher temperature, they have light brown points. So next, we'll talk about the influence of sex on the expression of alleles. First thing we'll talk about is sex-influenced traits. And I want to make the point that these are different than sex-linked traits. Sex-linked means that those genes are encoded on the sex chromosomes, the X and the Y chromosomes. And a lot of the time, sex-influenced traits look very much like they must be carried on the sex chromosomes. However, sex-influenced traits are generally found on autosomal chromosomes, and the expression of the phenotype is influenced by the sex of the individual. So here's an example in humans, hairy earlobes. For a long time, this was actually thought to be a Y-linked trait because it's only found in males. But recently, it's actually been figured out that this is an autosomal allele, and it is limited to males, however, so presumably due to sex hormone levels. I want to talk next about cytoplasmic inheritance. So this is a picture of a cell, and the DNA is stained, and you see lots and lots of DNA in the nucleus. However, there's actually DNA in the cytoplasm as well, and that is because mitochondria and chloroplasts have their own DNA. Cytoplasmic inheritance shows a really interesting expression pattern because it is inherited maternally. And so every cytoplasmic chromosome that you've got in your mitochondria came from your mother. So you didn't get any of those from your father. It turns out that there are actually some cytoplasmic disorders, and if you get this from your mother, 100% of her children are going to be affected because the mitochondria in the egg are passed on to all of the children. And so you see all of her progeny, both males and females, are affected. Now, males will not pass that on to their children because they just contribute a sperm, and actually the DNA in the mitochondria from the sperm get completely degraded once the egg and the sperm fuse together. I just want to make the point that organelle chromosomes are present in many copies per cell. So we really don't have this diploid versus haploid. We don't have, heterozygous versus homozygous, that kind of thing. There's lots of copies, and at a certain threshold, if you do not have enough good copies of a gene, you're going to show this mutant phenotype. In human, there are 37 genes on the mitochondrial chromosome, and here's a list of a number of disorders that are known to be inherited due to mutations on the mitochondrial chromosome. The last topic we'll talk about is something called genomic imprinting. Now, this is only found for maybe 100 genes in humans, and it is called monoallelic inheritance. So there are certain autosomal genes that the copy that you get from one parent is inactivated. So you only have one allele that is active. For maternal imprinting, the copy that you got from your mother is inactive, and so your good copy is from your dad. And then for paternal imprinting, the copy that you got from your father is inactive. So the way this happens is that during spermatogenesis or oogenesis, there are certain methylation marks that are put on the DNA that inactivate that gene in either the father or the mother. And that methylation remains on the DNA through the gametes and through those early stages of development. And in the progeny, all of the cells in that individual will have that same methylation pattern that they received from the gametes. So an example of imprinting is the IGF-2 gene in mammals. So this was first worked out in mice. And so the way this works is the IGF-2 gene is an autosomal gene. However, you just need one active copy in order to have a normal phenotype. So this gene is subject to imprinting, and it turns out that the copy you get from your mother is always silenced. So in a normal individual who has two good copies of that gene, the copy from the mother is silent, the copy from the dad is active, and you have the wild type, the normal phenotype, or the normal size of a mouse in this case. If you inherit a mutant version of the IGF-2 gene from your mother, it doesn't matter at all because the copy that you get from your mother is normally silenced, and so you still have that one good copy that's being expressed from the copy you got from your dad, and that is enough to make a normal phenotype. The problem arises if you inherit a mutant copy of the IGF-2 gene from your dad. So even if you have a good copy of the IGF-2 gene from your mother, this copy is always silenced. And so this one is silenced and this one is mutant, and therefore you don't have any functioning IGF-2 protein being made, and that gives a dwarf phenotype. So it was found actually that there's a human syndrome called Beckwith-Wiedemann syndrome that is caused by disruption of the normal imprinting of this IGF-2 gene in humans. This syndrome is a little bit different than the mouse example here because people with Beckwith-Wiedemann syndrome actually have lost their normal imprinting or their normal silencing of their maternal IGF-2 allele, and this actually results in an overgrowth phenotype in humans.